Have you ever walked through a forest and wondered, what exactly are you missing? Today in this explainer, we're heading deep into the tropical wetlands to uncover an entire world that is completely invisible to the naked eye, yet it fundamentally runs the ecosystem. We're not just looking at a dry scientific study today, we're actually stepping right into a biological mystery. Because for decades, researchers have basically walked right past a bustling, microscopic metropolis hidden up in the canopy and down in the understory of our forests. So who lives there? What are they doing? And perhaps most importantly, why have we been so blind to them for so long? So here's our roadmap for the explainer today. We're going to start with the hidden world of Hymenoptera, look at the baseline biodiversity, and then explore a tale of two traps. After that, we'll see exactly how behavior dictates detectability before wrapping up with the necessity of dual lenses. Section 1. The hidden world of Hymenoptera. Okay, so our setting here is the Tasek Bera Ramsar site over in Malaysia. Now, previous studies here focused heavily on the ground, and because of that, they completely missed the dynamic insects living way up in the canopy. Look at this incredible microscopic view of a Hymenopteran specimen. With those intricate wings, the specialized antenna, its whole unique morphology, these creatures are literally the hidden engineers of the wetlands, and they are notorious for being incredibly difficult to detect. Section 2. The baseline biodiversity. To even begin to understand this hidden ecosystem, researchers first needed a baseline, right? Like, who actually dominates this environment? Well, looking at the breakdown, Formicidae, which are ants, make up a massive 41.8%. But right behind them are the Echnumonidae and Broconata families. Those are your parasitoid wasps. Together, ants and parasitoid wasps make up over 80% of the collection. And this is a huge indicator of ecosystem health. The ants are acting as soil engineers, while the parasitoid wasps rely on other insects as hosts for their larvae, which keeps pest populations perfectly in check. Taking a look at the overall health, the researchers physically captured 67 individuals across 31 entirely unique morphospecies, which just means they look physically distinct from one another. But the absolute most crucial metric here is the high evenness. The community is evenly distributed. This means we aren't seeing an ecosystem being bullied or totally dominated by one single overly aggressive species. It's balanced. It's a highly diverse and robust ecological web. But here is where the mystery really deepens. 83.5. This is the Kakawan estimated richness. It's a statistical model that predicts the true number of species based on the rare ones we actually managed to find. So while researchers physically observed 31 species, the math is telling us there are actually around 83.5 out there. The big takeaway here is this massive knowledge gap between what we physically caught and the rare elusive taxa that are statistically still hiding from us out in the wild. Section 3. A tale of two traps. So how do we bridge that massive knowledge gap? It begs a really important question. Why does the type of trap matter so much in science? I mean, it sounds like a simple logistical choice, right? You just grab a trap and go. But it's actually deeply philosophical. In ecology, the trap you choose acts as a lens, and how you look at an ecosystem fundamentally, entirely changes what you end up seeing. To map Tasek Bera, researchers used two very distinct lenses. First, we have Malaise traps. You can kind of think of these as giant nets in the sky. They use active interception, so when highly mobile flying insects hit the mesh, their instinct is to fly upward, straight into a collection bottle. Then we have yellow pan traps. These are small bowls placed near the ground, filled with soapy water. They rely on visual attraction, using bright colors to mimic floral resources and lure in foraging insects. And when you use two totally different methods, you get totally different results. Check out these actual specimen photos from the study. From a ground-dwelling ant to actively flying parasitoids to tiny cryptic wasps. These distinct traps are capturing creatures with vastly different shapes, wing morphologies, and flight capabilities. It's like they're sampling completely different worlds. Section 4. Behavior dictates detectability. Now, this is where the researchers had a major aha moment. They realized the variation in their catches wasn't just random static. The traps were successfully profiling two entirely different neighborhoods within the exact same forest. Malaise traps were capturing the transient community. These are the highly mobile parasitoids traversing broad territories looking for hosts. They're basically the commuters of the insect world. Pan traps, on the other hand, well, they were capturing the resident community. These are localized, site-specific foragers tethered to immediate floral resources right down there in the understory. When we actually compare the sheer numbers, the Malays trap edged out with an abundance of 37 individuals, compared to the Pan trap's 30. The Malays traps essentially cast a wider net, pulling in a higher sheer volume and showing a much greater potential for discovering those rare transient taxa. But hey, the Pan traps shouldn't be underestimated. They provided a much tighter, incredibly even representation of the immediate localized community. They were equally effective, they were just measuring different things. Section 5. Necessity of dual lenses. All right, it's time to synthesize this detective story. If you take away anything from this explainer, I want you to remember this quote. Trap bias is not a methodological flaw. It is an ecological lens. Seriously, this is a massive paradigm shift. If you go into a forest and only use one type of trap, you might walk away thinking you've surveyed the whole area. But the reality is, single method surveys completely misrepresent ecological reality. You're only ever seeing the creatures whose behavior perfectly matches your specific trap. So how do we fix this going forward? Well, the researchers laid out 3 really clear, actionable steps for future monitoring. First, temporal expansion. We need a sample across both wet and dry seasons to account for floral blooming cycles. Second, spatial stratification. We absolutely must systematically pair canopy traps with ground traps to map out the vertical layers of the forest. And third, molecular resolution. We need to integrate DNA barcoding to rigorously identify those identical-looking cryptic wasps that traditional microscopes just can't tell apart. You know, we started this explainer looking for invisible bugs, but we ended up learning a whole lot about human perception. Our view of nature is entirely bound by the tools we invent to observe it. By embracing complementary, multi-trap approaches, we get a little closer to the truth. But it leaves us with one final thrilling question to chew on. If we only see what our current tools allow, what else is still out there, completely unknown, just waiting in the shadows of our forests? Thank you so much for exploring with me today. Keep asking questions and keep looking closer.